Thanks to everyone at Cavendish for a terrific conference and for this opportunity to speak to you. So who here in the room would be interested in curing cancer? A few? Okay, good. Well, hopefully you're going to see something intriguing here. There are a number of different cancers. These are sort of the big ones that everybody has heard of and that affect a huge number of people. And they have a lot of different things in common. One is all the ones on this list are what are called epithelial cancers. So they start in the membranes of different organs or essentially in the epithelial layers of your body. They have other things in common, of course, and they strike over 1 million people in the United States every year, and they kill hundreds of thousands of people. About 300,000 people in the United States will pass away from these cancers every year. They have one very important thing in common, and that is this cell. So about 6% of your circulating T cells, T cells are the cells in your bloodstream that eliminate pathogens. Well, it turns out you have T cells in your body that also eliminate cancer. And one of those types of T cells is this V delta 2 gamma delta T cell. Now, it's highly effective at this, and I can guarantee you that every single person in this audience has some level of epithelial cancer, and it really is completely unimportant because these very small occurrences of the epithelial cancers are very easily eliminated by these gamma delta T cells, and the reason that you have so many of them in your body is because they are constantly surveilling and constantly eliminating these 1 or 2 cell occurrences of these epithelial cancers, and that's why it never develops into what we call a cancer, which is a solid tumor. The problem comes that as the solid tumor grows, you can imagine if this was a balloon, the outside of the balloon is getting quite big as it grows, and the gamma delta T cells can't keep up with it. Now, what we discovered is that we can drive these gamma delta T cells, these natural T cells, to 300 to 600 times their normal activity. So they can go ahead and they can chew up a solid tumor. The way we do that is because they have this T cell receptor here, which is like the gas pedal for the system. It isn't used in recognizing the cancer cells. But what it does do is it causes them to activate and it causes them to proliferate. So we can raise the number of these cells in your body, and we can also raise the activity of each one of those cells. And then they recognize with that NK receptor. That NK receptor is extraordinarily specific to diseased tissue. And this is very important because there are therapies out there, you've probably heard of CAR T, where in some cases, T cells can be driven to the point where they start to eat the wrong things. Well, it turns out that gamma delta T cells have never been associated with an autoimmune disease. And what this means is that they've never misrecognized a tissue and they've never eaten the wrong thing. So how do we use these gamma delta T cells? What's our process for eliminating cancer? Well, when you have a solid tumor, it looks like normal tissue to most of the body, and it actually can repel immune cells. As a matter of fact, when you hear the word immuno-oncology, generally what you're hearing it applied to is either CAR T or these new drugs that block what are called checkpoint molecules. You'll probably hear the word checkpoint inhibitor. Well, cancers can actually put up fake little tags on the outside of themselves that make them look like organs. One of those common tags is called PD-L1. PD-L1 is also used on your heart. It also is an extra added protection that your T cells will never go after your heart. And in very rare cases, when you block those PD-L1 with a monoclonal antibody, so Keytruda, which is a miracle drug, basically blocks PD-L1 all over your body. In rare cases, it blocks enough of it on the heart that your T cells can end up going after your heart. And in those cases, people will die in less than 3 days. That's how quickly T cells can eat tissue. So think about the power of that. Now, the gamma delta T cells don't have the problem of misrecognizing any tissue. They're not looking or being blocked by these PD-L1 molecules or other checkpoint molecules. And also, they won't end up misrecognizing cancer versus a normal tissue. By the way, the NK receptors have, the way that they recognize it is, there is a metabolic state when these cells start to rapidly divide, and that causes some signaling inside the cells that the NK receptor can actually click onto and turn on or off the cell killing based on seeing that. That's why they're so good at identifying cancers. So what we do is we take the solid tumor and we can reprogram some of the cells in there to create a stimulatory peptide, and I'll explain that in a minute, that pushes down on that gas pedal. Now remember, by raising that stimulatory peptide, we're not signaling that this is a cancer cell, even though we're doing it in cancer cells. What we're doing is it's like hanging a neon sign on the solid tumor. It's like a sign that says free lunch, all gamma delta T cells invited. When they get there, they start recognizing the cancer cells and they activate. And when they activate, that's when they go into this stage where they're eating at 300 to 600 times their normal rate. So the activated T cells will immediately destroy the primary tumor. And to give you an example of how effective that is, in a 1,000-mouse study, we're getting 80% complete remission in a variety of epithelial cancers. So breast, prostate, liver cancer. These are some of the ones that we've tested. And 80% of the mice have no cancer left at the primary site. And that would be the place where we are actually injecting this virus. It's actually a virus that reprograms some of those tumor cells to emit this peptide. That tumor goes away completely. But here's the side benefit. These activated gamma delta T cells continue to circulate around the body through the blood and the lymph systems. And what they're passing is the metastases and the secondary tumors. They're still in that activated state. So what we're seeing is what's called an abscopal effect, where it's clearing secondary tumors and metastases. So we believe that this will be effective even in late-stage cancers. This is what I was talking about with the mice. So see where it says control there? That's untreated mice. So you give them these cancers and they expire after about 50 to 60 days. So the cancer gets so large that the mice are suffering and they have to be euthanized. When we put the vector in, so we reprogram the cancer with our vector, what happens is it starts to make these stimulatory peptides, and this takes up some of the metabolic energy of the cancer, so it slows it down a little bit. But it's not curative. What happens is they just live a little bit longer. When we introduce natural gamma delta T cells from human monocytes, so the gamma delta T cells that are natural in your body, you get cures. So you're getting 80% of them with no cancer left afterwards. I left this slide in because I saw some of my colleagues had already prepared you with some fairly gross slides. But what we did was we went ahead and on the mice, we grow a centimeter tumor. That would be a huge tumor in relation to a human. That would sort of be like having a tumor the size of a grapefruit on your body or even larger. Then we go ahead and we inject the human monocytes into the peritoneum. We need to make sure that it's not near the tumor. We need it to seek out the tumor, and we need to show the efficacy that way. Well, that's what we got, is these mice over on the right here were the treated ones, and they got, all of those ones had no tumor left in them. This one here, we actually think what happened is called tumor lysase syndrome. So the tumor may have been so large that it broke up so quickly that it was the cell debris poisoned the mouse. So in any case, untreated, you can see the size of the tumors over there, and treated mice had a startling result. So let me back up a minute and tell you a little bit about American Gene Technologies, because we're really broader than cancer. We ended up in cancer because we found that we had some technology that was efficacious in that. But the company started about 10 years ago. I had a background in computer science. I'd founded a number of different companies in Silicon Valley, and after I retired, I had some extra money and time, and I didn't want to go back to 15-hour days, but I thought I'll just go ahead and do some angel investments, and I started getting business plans, and I met a scientist at National Institutes of Health. His name's Roscoe Brady, and he showed me viral vectors. So viral vectors are basically, we can now break open viruses, deadly viruses like even HIV, and we can scoop out the viral DNA, and we can replace that DNA with whatever we want. Now, what do viruses do in your body? They basically hijack your cells by bringing little programming code, essentially code segments into your body that hijack the cells. And it's useful to think about your cells as organic computers. And let me see if I can draw a parallel that you can understand. So the computer on your desk or in your pocket or your laptop or whatever, the software is all coded in zeros and ones, binary. You've probably heard that before. And it forms what's called an operating system. So you have the Microsoft operating system or the Apple operating system, and this is just a set of tools that app developers use to write things like Word or Excel or whatever. But the operating system is essentially the DNA of your desktop computer. Well, the DNA of your human computer is actually DNA. It's coded in 4 amino acids, A, C, T, G, and it's the order of those acids that determine everything that the cell does. So if you go ahead and you change what's in the DNA, the cell changes. We've known this for many, many years. And this is exactly what viruses do, and they can actually hijack your cells to do very complex things, including replicating viruses. Now, in the case of HIV, it doesn't really do a lot of replication of itself. It replicates very slowly, but it's a very clever virus because it actually plugs new genes right onto your original DNA, and it makes them permanent changes to your DNA. So what did we do with the lentiviral vector? We crack open HIV, we scoop out AIDS and throw it away, and then we can put code segments in there that convert that virus into an update, something that makes your cells operate better. These updates can even do things like clear viruses. You can even cure HIV with that. They also can repurpose cells, like in CAR T, to go out and hunt down other cells, or repurpose cells in your body to create these stimulatory peptides to cause an immune reaction. But the basic idea is that AGT is about reprogramming the human computer and the amazing amount of things that can be done with that. It's an incredibly powerful technology. It fixes things from the inside out as opposed to putting chemotherapeutics all over your body that don't know where to go. These things can be targeted to only attach to certain cells and only turn on when they sense different disease states. They have like if-then commands inside this software, sort of like a smart fuse that turns on the medicine, but only if it sees the disease state or only if it sees that it's a certain type of cell. So that's what AGT is about, is leveraging that. So we have a very powerful operating system, and we intend to be the Microsoft or the Apple iPhone of this industry. What we want to do is make it really simple for people who have knowledge of certain diseases to go ahead and cure those diseases using our tool set, be able to reprogram the human computer to get value out of it. And we have 28 patent families, and we've already gotten valuations on our IP of $1.7 billion worth of IP. We've only spent $17 million to date, so that's very efficient. And we were in an area of discovery where there was a lot of white space. So we've managed to make a lot of fundamental improvements to how viral vectors are used, including constructs that can go in the viral vectors to do useful things. So we can end up with a machine like Microsoft that could start just turning out app after app after app. But how would you fund that? Well, I'm sure you're all thinking the same thing as I am. You'd cure HIV. We're breaking open HIV, and it turns out that that's actually a really good target for us. Now, the cancer stuff that I showed you is going to take us till about 2020 to get it into the clinic. And we're collaborating with Stanford University, a guy named Dean Felcher there. He's an expert in hepatocellular carcinoma. Liver cancer is a great first target because, unfortunately, almost everybody who's diagnosed with HCC dies of HCC within a year. So the FDA is fairly open to trying new things. And so that will be our first test of this epithelial cancer drug. But if it works on that, it can be spread very quickly to prostate and breast and all these other cancers. We can prove it's safe in liver cancer, and then we can move on to other cancers. But to drive this development, we have a project right now that is going into the clinic this summer that is a cure for HIV. And the way that it works is we go ahead and we pull blood out of an HIV-infected person, and these people have to be controlled on antiretroviral drugs. You know that when you're on an antiretroviral drug, if you have HIV, your immune system is restored. That's why you stop getting colds, flu, you don't die of pneumonia or something like that, because the antiretrovirals hold down the HIV infection low enough that your bone marrow and your immune system will recover. So now you have a full set of T cells. And what we do is we take 400 milliliters of blood, we isolate the HIV-specific CD4 cells. These are the ones that drive the whole immune reaction to HIV. And then we protect those cells against HIV entry. Because it turns out the only reason that HIV gets a hold of your body is it's got one benefit over other viruses. And that is, when that HIV T cell comes along and attacks that viron and tries to kill it, turns out the viron can just merge right into the T cell, attach its DNA to the T cell's DNA. That T cell gets excited. It goes to your lymph nodes and starts replicating. It actually becomes the beachhead for the disease, instead of that sentinel cell doing what it's supposed to do. If this was a flu viron and a flu T cell, you might only have a handful of them in your body. The first one meets that viron, it reliably kills it. It can't merge. It kills that viron, and then it gets excited, and it goes and gets friends and comes back and kills some more, and they get friends, and pretty soon, if you didn't stop that replication, you know your T cells would actually replicate so quickly that your body weight would double in 3 days if that reaction didn't stop. But you can get on top of the cold that fast. But in HIV, you could do the same thing. It's actually a slower moving virus. But the problem is you have to stop that merge. Well, what we do is we reprogram those HIV-specific T cells so that they don't have the handle that HIV grabs onto to merge. It's called CCR5. So HIV can no longer get into the T cell. It can no longer deplete that T cell. That T cell operates normally, and it clears the virus. So what happens is that after 12 days of treating their blood, we reinfuse it in. Within a week, they can go off of their antiretroviral meds. They can never develop AIDS. They can never give HIV to another person. They're not contagious. They'll eventually completely clear HIV from their entire body, and they can never be reinfected, ever. So they're better than cured. They're now immune. Because it turns out what we've given them is a mutation that is actually natural in a very small percentage of people in the world. There are rare people of Northern European descent who have something called a delta 32 mutation where they're missing CCR5 on all their T cells. And it doesn't hurt them at all. They're walking around with basically normal lives, but they cannot be infected by HIV. And the only reason is that HIV cannot merge and deplete those sentinel T cells. So we're essentially making mutants. But in the good way, not in the X-Men or whatever sense of the word. So anyway, that's going to come out. We're going to show first human efficacy in that this year or in the first quarter of next year. That is a $15 billion asset. That is actually, if you take our current valuation and our current round, that's nearly 100 times the current market cap of our company for that one drug. And what we'll do is we'll use that as a way to buy back stock. We won't even need to go public. We'll use that money to fund this process. Every time there's good data about what the genetic underpinnings are of a disease, and we can think of a creative way to mitigate that disease by knocking down a gene or overexpressing a gene or reprogramming a cell in some way, then we can develop that to first human efficacy in 2 years and $10 million per project. So you could see that we could have a huge number of things coming off this assembly line. It gives us the opportunity to study all different types of diseases, the key thing being that we understand the genetic underpinnings of it. And guess what? That's what the entire research community is giving us every day. They're publishing papers every single day that explain to us which things are driving which diseases. And it gives us the ability to experiment. The other thing that's amazing about doing genetic medicine like this is because it's so targeted, it works the same in the human as it does in the mouse. In other words, it doesn't fall out because it causes some side effect in the liver. It doesn't even turn on in the liver. So because of the targeted nature of this, because the gene is the same in the mouse and in the cat and the dog and the monkey and the human, it translates beautifully. So most of these drugs, it's probably 1 in 5 of these drugs will make it out to commercialization. So this has tremendous potential. This whole industry has tremendous potential, and there's other gene and cell therapy companies out there, but we're the only ones who said this is the first time where a platform is critically important to getting the power of gene and cell therapy to the most people at the fastest rate. And that's what we're committed to. We work with researchers from top-notch universities across the country. This is just a small sample of them. I wanted to show you this one because John Rossi is our collaborator at City of Hope on the HIV project. We just lost Bob Redfield, who's going to head up the CDC now, but he was our lead person on the HIV project up to now. John Rossi's amazing. Dean Felcher, I mentioned to you from Stanford. Jerry Vockley is heading up our Phenylketonuria drug, and he's at the University of Pittsburgh Medical Center. Ted Dawson is working on it with us on Parkinson's. He's at Johns Hopkins. And Robert Clark will be working with us on breast cancer once we can get an initial proof of concept in liver cancer. We have an experienced executive team and just over 20 people, most of them in the R&D department, in a facility in Rockville, Maryland. And if you want to hear a lot of hope for a lot of different diseases, talk to us. And if you're interested in helping us propel it, join us. Thank you.
Thank you, Jim.